So hi everybody. Special thanks to the organizing team. Thank you. So today I'm going to talk about sugar beet. So I'm Augustin Desprez. I'm currently at the end of the second year of my 3-year PhD. And today I'm going to present how three-way hybrid breeding have impacted structuration and diversity of breeding material. So within one company germplasm, because each company has its own germplasm in sugar beet, very different. So, and how here we tried to identify selection sweeps specific to the differentiation between pools. So first, let's just quickly recap the sugar beet. I'm sure you are all very, very familiar with it. So the context regarding breeding strategies in the past decades with their impacts on yield, for example. So here you can see that for the first period here, we are breeding populations, and we have really a break into the slope coming here from two innovations that led to the current way of breeding, which is three-way hybrids. We also have several innovations with introduction of traits coming from the crop-wide relatives, so beta maritima, with tolerances that are well known. Of course, you are aware of a lot of disease management in sugar beet, I'm sure. And today I'm going to present you the results from a panel of 20 years of elite germplasm, so that are located here the past 20 years with 3 breeding periods. So just to quickly explain how did we came up with three-way hybrids in sugar beet. So first, sugar beet breeders were facing the fact that there is a lot of inbreeding depression, so they were of course interested in hybrid breeding, and it has been clearly explained with maize results during the 1930s. And to do so, they had to use cytoplasmic male sterility because castration was impossible to do on a large scale. However, this kind of sterility requires O-type and maintainers. So O-type is specific to sugar beet, but it's just sterility maintainers. And on the other hand, they were also facing the fact that seeds at that time required a lot of manual thinning. So each fruit presented multiple seeds, which required then a lot of manual labor. So another discovery, the discovery of monogerm fruits, a single recessive gene, made the fact that it was possible to have monogerm seeds. However, it reduces a lot the seed production. Therefore, they came up with this breeding pattern. So here you can clearly see the female pool, which is hybrids. So why having female hybrids? In order to increase the seed production of the female seed bearer here, called MSF1. And also on the other side, they have male pool pollinators that are inbreds in order to produce the three-way commercial hybrid. So coming up with this, we asked ourselves several questions. So the first is, after 60 years of three-way hybrid breedings, what are the impacts of three-way hybrid breedings, both on genetic diversity and on genetic structure? And also, today, can we identify genetic patterns of selection, so selective sweeps along the genome that are linked to this hybrid breeding? So I'm using 10,000 SNPs and a wide panel of accessions. So as you can see here, because of the asymmetry in the breeding panel, we have a lot less female parents than male parents, but it's linked with the technical constraints of having to manage cytoplasmic male sterility with longer schemes on cycles on the female side. So really, if male and females are absolutely not breeding, the breeding methods are very different between the pools, and always they are evaluated regarding the vigor of the commercial hybrids. And here you can keep in mind the colors that I'm going to use throughout the presentation. So first, just looking at global diversity. So here with the PCA and the SNPs, so you can clearly see the differentiation between male and female. Females on the left side, male on the right side. And here also you can see that males are colored with the years of the pollinator. So older pollinators at the top in green and newers at the bottom in red. You can clearly see that there have been a shift of structuration during the years following this trajectory. Looking at other PCA axes, you can see, of course, because of the number of accessions, a lot of structuration within the male pool. Again, newer accessions, older accession, and in between. And clearly, the same structure is appearing when regarding admixture. So here, creating two ancestry groups. So you can see the females here are associated with the yellow group and male in the purple group. And when you increase the number of ancestry group, you can see that there is structuration within the male pool. And this structuration here, when you color the individuals with colors, with the year of the pollinator, you can again see that the structuration is highly linked with the year. So this is why we came up with looking at 3 eras, 3 breeding periods within males. So as you can see here, the older and the intermediate and the newer breeding periods matches pretty well with structuration. So here, looking at diversity, so here I'm using MRD, so pairwise genetic distances between individuals. And here you can see that globally we have quite a high values of MRD, but also we have the evolution within the male pool. So a slight decrease in diversity within males through time, which is quite expected because of the high selection thresholds. And looking here at FST, which represents the allelic differentiation between male and female hybrids. So here each value corresponds to one marker and how the allelic frequencies are different between the two pools. And we can see here that we have very, very high FST because the average is around 0.25. But also we have a slight increase here, and you can clearly see it when looking at the distribution. Like the bottom of the distribution is very reducing throughout times, and the mean and median are increasing. So in 20 years, plus 12%. So we can see here that there is maybe a link between the fact that we are selecting and we are increasing the differentiation between the two heterotic pools, the two male and female pools. And looking here across all 9 chromosomes, so you can see at the top the genetic diversity within males that is evolving, and at the bottom the differentiation between males and females. So here you can clearly see the link that a decrease in diversity can lead to an increase in differentiation, of course, which means that breeders are maybe increasing globally the divergence, so maybe there are a lot more hybrid vigor within commercial hybrids. And here, just looking at genome-wide values, again, across all 9 chromosomes. Here, the gray region are the centromeric regions. And here you can see, so allelic differentiation again. And you can see that there are a lot of discrepancies along the genome. You can see, for example, very, very wide here patterns in centromeric regions, of course, because of low recombination. But you can see that some regions have experienced a very high decrease throughout the 20 years. So green at the top, red at the bottom means a decrease. And this can be linked with several breeding methods. For example, using the same founder in both pools, introducing, I don't know, a disease resistance from the same donor in both pools, so bringing them together in certain location. But you can also see that we have a lot, and in fact more, because we saw that on average we have an increase in differentiation, but more patterns that are linked with an increase in male and female differentiation, which of course can be linked with breeding for heterozygous in hybrids, so for example breeding dominant alleles or general vigor. And also an increase of differentiation can come from drift because those pools have a very, very low level of genetic flux between them. So now we are interested in identifying if we can see specific regions that are linked with differentiation between male and females. So let's look at it. So in order to do so, we used two methods that we combine in order to limit the identification of drift-linked patterns. So the first method was using PCA DAT. So we are using here the first axis of the PCA, and to know which markers are contributing the most to this axis. And the second method was using FST, so allelic differentiation. And this is the kind of patterns we get. So here, FST on the y-axis and the p-value associated with contribution to the first PCA axis. And with this, we can identify 174 loci that are in common between the two methods. And we can clearly see when we are looking at the 9 chromosomes along the genome that maybe we have clusters of differentiation. And for example, zooming in the second chromosome, you can clearly see that we have a pattern here that is really coming through. Maybe the same demographic history is shared within this location. And in this location, it appears that we have a domestication gene, which on first sight is quite surprising because it's fixed on both pools. However, we still have a lot of differentiation, so we still need to clarify if it comes because this domestication appeared using 2 different founders, or if it's due to later breeding consequences. And here, you are looking at patterns of selection. Colored blocks, so we are looking at linkage disequilibrium blocks across all 9 chromosomes, so haploblocks. And the y-axis is the number of haplotypes, so kind of a diversity index of the block. And the colored blocks are the ones that carry selective sweeps. And what we see here is that with 174 loci, there is only 30 haploblocks, and in fact, it's only 7 clusters of candidate regions that are carrying 60% of candidate SNPs, so very, very high here in yellow, for example, or the chromosome 2 signals that I just showed you is here. So this is exactly what we see here. So just looking, for example, at this haploblock here, which carry 45 SNPs and has very, very high FST, very, very high differentiation between heterotic pools. Here, if we look at haplotypes and how they are distributed among pools, so we can clearly see here. So the first 2 haplotypes are the ones that are carried by the males. And here you have a few examples of haplotypes carried by females. And you can clearly see that looking here at these columns, which are the frequency of the haplotypes in each pool, we can see that the first one here is inexistent within females and has increased a lot during the 20 years time period from 37% to almost 100%, so 85%. Of males today that carry the same haplotype. So clearly we can see that here the selection have had an effect in the past with a clear differentiation between female and male pools, but also we can see that it's still occurring today with very, very high selection rate within male and females, and also we can identify where in the haplotypes the selection occurred with the specific sub-regions. So clearly, this 20 years time period is enough to see very, very strong shifts in structuration and differentiation within an heterotic pool. So to sum up, we've clearly seen that genetic diversity within male has slightly decreased over time. However, it seems that it has contributed to differentiation. And also we saw that 3-way hybrid breeding has shaped sugar beet germplasm. So here, we just saw the example of one germplasm. It's maybe not the same in all companies and even in programs within companies. So very, very high differentiation, but it is very different along the genome with specific regions that contributed to hybrid breeding. And also, we will in the future publish this in an article next year and also have a look on how to increase breeding methods. So thank you.